Blood. How will you describe blood or the properties of blood, functions of blood? Anyone?
Contains red blood cell that carry oxygen.
Okay, good. It contains red blood cell that carries oxygen. Okay. Anybody else?
Carry nutrients.
Carry nutrients. Okay, correct. Then,
It has also contained the hemoglobin. It looks like the red.
okay, correct. Anything else? Okay, anyway, we will discuss its function, its components, everything today. So today's lecture will cover the functions of blood, the composition of blood, that is how the blood is composed of, what it is made of. Then blood plasma, then formed elements. This is the, I mean, formed elements are elements which are present in the blood. Then blood transfusion, how the blood is transferred from one person to other, the blood groups, all those things. So these are the topics that we are going to cover today: functions of the blood, composition of blood plasma, formed elements, and blood transfusion. So these are some of the general properties or aspects of the blood. We call blood as the river of life, and blood is also the only fluid tissue which is present in the human body. Generally, tissue is like a solid mass, but blood, it is the fluid tissue. So we call blood as the river of life. Its role is to circulate and present throughout the body. So wherever you get a cut, you get a wound, or you get an injury, either it could be at the top of your head or at the bottom of your toe, you can see the blood. So in general, the blood is bright red in color because it contains oxygen. Whenever the blood is rich in oxygen, it will be in bright red color. Due to some situation or due to some diseases, some people, the blood inside some people may not carry optimum level of oxygen. For those people, the blood will look like dark red color, or maybe dark brown, something like that. But in general, dark red. So if the blood is rich with oxygen, it will be in bright red. If the blood is low with oxygen content, then it will be dark red. But the blood will never look like blue color, because sometimes when we look at the blood, we can see vessels which has which looks like blue color. So some people may think blood is blue, but no, blood can never be blue color. Then the pH level of the blood is around 7.35 to 7.45. Does anybody know what is the meaning of pH? Ian,
Acid and
sorry?
the acid and alkaline level.
Yes, one extreme is acidity, another extreme is alkaline. But what does the pH stands for?
Potential of hydrogen.
Yes, the hydrogen content present in the liquid, or if you are talking about water, then it is. Since we are talking about blood, so the level of hydrogen present in the blood. So in our blood, the hydrogen level is around 7.35 to 7.45. So that is what we call it as pH. Then the average temperature of our blood is around 38 degrees Celsius. It is around 38 degrees. It's not like exactly 38 degrees. Sometimes it can go for like 38.4, 38.5. Sometimes it can go lesser, like 37.5, 37.8, something like that. So it is always around 38 degrees Celsius. Then the volume of blood that is present in our body can vary based on the age, based on the sex, and based on our body size. So depending on these three factors, the volume of blood may vary. But still, there is an average value. We will discuss that in the further slides. Next is functions of blood. So these are all the possible functions that a blood can do. So blood helps in maintaining the homeostasis. In last semester, we have discussed about homeostasis. Does anybody remember homeostasis, or does anybody know what is the meaning of homeostasis?
regulating our body temperature so that it adapts to the outside environment.
Sorry, can you repeat again?
Homeostasis is the condition that our body regulates our body temperature to adapt to outside temperatures in the
Ah,
environment.
okay, some sort of stability or regularity, to simply put. Anyway, fine. So the body helps in maintaining homeostasis by doing the following functions. One is transporting the gases, nutrients, and it is supposed to be waste. That's a mistake. Then transporting processed molecules, then transporting the regulatory molecules, then regulating the pH level, that is the potential of hydrogen that is present in the blood, then regulate the osmosis, then maintain the body temperature, and to form a clot. So these are all the possible functions of blood: to transport gases, to transport nutrients and waste, transport processed molecule, transport regulatory molecule. Does anybody know the difference between processed molecule and regulatory molecules? Processed molecules, regulatory molecules. Anyone?
Process is like change from one state to another state, while
Uh-huh.
regulate control the amount. Is that correct?
Can you repeat the regulatory molecules?
Regulating is like control the amount from the last number to a higher amount.
Uh, uh, I'm not able to hear your voice clearly. But anyway, regulatory is like we have some hormones which are secreted at one part of our body, which can regulate the function of some other organ or some other tissues. So the function of blood is to transport those regulatory hormones or regulatory molecules. Those things comes under regulatory molecules. Then pH, as we discussed earlier, it is the level of hydrogen present in the blood, and regulate the osmosis, maintain the body temperature, and formation of clot. So these are all the functions of blood. Next one is composition, how the blood is made up of, what it is made up of. So blood is a type of connective tissue. Last semester we studied about connective tissue. So blood is also a connective tissue. It is a type of connective tissue. It consists of a liquid matrix called plasma, which has cells and cell fragments. So plasma consists of the cells and the fragments of the cells. This plasma makes up to 55% of our total blood volume, and the formed elements, it makes up to 45%. So 55 plus 45 equals to 100%. So there is a test called centrifuge test. So whenever you donate your blood, I'm not sure how many of you have donated your blood. So anyway, so whenever you donate your blood, the hospital or the lab, they will process your blood through this test called centrifuge test, where they can separate the plasma, the WBC, RBC, the platelets, everything. So that test is called as centrifuge test. So after doing the centrifuge test, we can easily see that the plasma makes up to 55%, and the formed elements form up to 45%. So the total blood volume in the average adult is about 4 to 5 liters in the females and 5 to 6 liters in males. This blood makes up to 8% of the total weight of our body. So if your body weight is 100 kilograms, then 8 kilograms is contributed by the blood alone. Next, this is what I mentioned, the centrifuge test. So here a person is donating the blood. After centrifuge test, this is how it looks like. So at the top, we have a transparent fluid-like thing that is called as plasma, which contributes to 55%. At the bottom, we have the formed elements. So again, this plasma consists of proteins, water, and other solutes. Whereas the formed elements, it consists of platelets, WBC, and RBC. And these are all the sub-classifications of the plasma and the formed elements. So we will be discussing about this in the upcoming slides. So this is how it looks like, our blood after undergoing the centrifuge test. The plasma will be on the top. It's like, imagine that you have mixed, you have a glass, transparent glass, and you have oil and water inside it. You just mix it. After some time, you can see the oil is on the top and the water is on the bottom. Likewise, here also, after the centrifuge test, the plasma will be on the top and the formed elements, that is the platelets, WBC and RBC, will be at the bottom. Next is plasma. So plasma is the liquid part of the blood. It is actually pale yellow in color. It consists of about 91% of water and 9% of other substances like proteins, ion, nutrients, gases, all these things. So based on the molecular size and charge, the plasma proteins can be further classified into albumin, globulin, and fibrinogen. So these are the three components that are present in the plasma: albumin, globulin, and fibrinogen. So we are going to discuss about the albumin, globulin, and fibrinogen. So albumin. So albumin is important in regulating the movement of water between the tissues and blood. The line is self-explanatory. Uh, because the albumin does not pass easily from blood into the tissue. So every tissue has the plasma membrane. The plasma membrane acts like a layer or the entry point for the other substance to enter. It could be either a nutrient, it could be a water, or it could be ions. Here, the albumin cannot easily pass through the tissue. That's what here it is represented. This albumin plays an important role in maintaining blood colloid osmotic pressure. So there is a pressure called as BCOP, blood colloid osmotic pressure, which is present inside our blood among the tissues. So this albumin plays a major role in this BCOP. Albumins can also bind and transport other molecules in the blood, such as fatty acids, bilirubin, and thyroid hormones. So this albumin can also, along with the other plasma things like globulin and fibrinogen, it can also transport some fatty acids, thyroid hormones. So anyway, the main function of the albumin is to regulate the movement of water between the tissues and the blood. So this is the primary function of albumin, regulating the movement of water between the tissue and the blood. Next is globulin. So globulin is again further subdivided into alpha, beta, and gamma. So this globulin function, the function of globulin is transporting many substances in the blood. They protect against microorganisms. So this is the primary function of globulin. They protect against microorganisms. Next is fibrinogen. This fibrinogen is responsible for the formation of blood clot. So each and every subdivisions of plasma has their own individual role: albumin, globulin, and fibrinogen. For albumin, it regulates the movement of water between the tissue and the blood. For globulin, it protects against the microorganism. Then fibrinogen, its role is to form the blood clots. Whenever we get any injury, any cut, the fibrinogen which is present in the blood will try to form the blood clot. So these are the main functions of this plasma proteins: albumin, globulin, fibrinogen. Clear? Serum is a plasma without the clotting factors. Okay, if there is no clotting factor present, then it is called as serum, not plasma. Plasma contains the clotting factor, that is fibrinogen. If this is not present, then it is simply called as serum. Serum is nothing but a plasma without clotting factors, that is without fibrinogen. Next is solutes. Here we saw solutes, right? So we saw about the water. There is nothing to explain about the water. We saw about the plasma proteins. Next is solutes. Solutes are nothing but ions, nutrients, waste, and gases. So ions like sodium, potassium, calcium, magnesium, and hydrogen ions, they are present in our blood. Nutrients, we have glucose, amino acids, cholesterols, and vitamins. They are called as nutrients. Again, that are also present in our blood. Then waste products like uric acid, amino salts, and creatinine. These are all the waste products which are also present in our blood. Then gases like oxygen, carbon dioxide, nitrogen. So these are all the gases that are present in the blood. We know about oxygen, carbon dioxide, but why nitrogen? Does anybody know the uniqueness of nitrogen? You could have heard of nitrogen somewhere else as well. Any idea?
Doctor, does it involve with the acidity of our body?
Acidity of?
Our body,
Uh,
nitrogen.
you mean it can increase or decrease the acidity?
Yes.
No.
Okay.
Anyway, good guess. Anybody else? There is something special about the nitrogen. Anyone? Okay, let me give you a clue. Nitrogen is called as inert gas, INERT, inert. Do you know what is the meaning of inert gas? Okay, let me ask another question. You could have noticed like syrup or jam bottles, wine bottles, alcohols, those things comes with the glass bottle. They are always inside a glass bottle, not in a plastic bottle. Anybody know what is the reason?
Doctor, about the
Yes.
inert gas,
Uh-huh.
uh, I think inert gas is the gas that already achieve uh, something like stable.
That's it.
I think stable,
Yes.
stable something.
Yes. That gas is stable. It does not interact or react with the surrounding. Say, for example, if iron rod is exposed to oxygen, after some period of time, that iron rod will become rust. It will get some rust because it is interacted with the or it is exposed to the oxygen. Likewise, there is a gas called nitrogen. It is an inert gas. That means it is very stable. It will not interact with any other surrounding structures like the glass. You know why? What is the reason for people to use glass to store the wine, to store the syrup or jam? Because glass is like it is like inert gas. Glass is not a gas, but assume or imagine it like inert gas. The glass will not interact with the surroundings. That is the reason why they store alcohols, wines, what else? Syrups in glass bottle. If they store it in a plastic bottle or something else, it may react. So within a few days of time, that product may be expired. So that is the reason they use glass bottles. So likewise, nitrogen is inert gas. It does not interact with any other surroundings. So nitrogen is also present inside our body. So these are the three gases: oxygen, carbon dioxide, and nitrogen. So these are all the solutes that are present in the plasma, which is present in our blood. Next is mechanism. It is like a general mechanism, nothing to, there is no need to give more importance, just for general knowledge. The amount of water taken in through the digestive tract closely matches to the amount of water lost through the kidneys, lungs, digestive tract, and skin. Can anyone explain this first sentence? The amount of water taken in through the digestive tract closely matches the amount of water lost through the kidneys, lungs, digestive tract, and skin. What is the meaning of this sentence? Anyone? Anyone?
Junior.
Doctor,
Yes.
does it mean that the amount of the water taken is to replace the amount of the water that lost?
Uh-huh. Can you repeat again?
The amount of water that has
Uh-huh.
been taken
Mhm.
is to replace the water loss.
Is to, okay, somewhat close, but not very detailed explanation. But okay, anyway, good guess. Thank you. Anybody else? Anybody else would like to explain this sentence? The amount of water taken through the digestive tract closely matches the amount of water lost through the kidneys, lungs, digestive tract, and skin.
Doctor,
Yes.
so the amount of water taken in through the digestive tract means that the water that we drink through water
Yes. Uh-huh.
or the water from our food and the fruits that we consume.
Yes.
And it matches the amount of water lost through the kidneys, lungs, digestive tract, and skin means when we're sweating, when we're breathing, when we excrete liquids from our body, it's the same so that we don't become dehydrated.
Yes, exactly. So it's like X equals to Y, something like that. So whenever we drink water, it doesn't mean our body completely absorbs all the water content. So the kidneys, the lungs, the digestive tract, the skin, they all have their separate function. So the role of kidney is to purify the liquid content in our body. The skin, they try to sweat. The digestive tract, they also use some liquid to what to say to soften the waste. So some amount of, so imagine this is our body, and some amount of water is always stored inside our body. So because of the functions of the kidneys, lungs, digestive tract, and skin, the amount of water is reduced. So whenever we drink, whenever we eat any food or fruit, the water content from those things will replenish this water content which is present in our body. So it is always like X equal to Y. So the amount of water taken through the digestive tract is always equal to the amount of water lost. That's it. And okay, next is since plasma volume remains relatively constant. So that is the reason why the plasma volume which is present in our body is always constant. The dissolved substances in the blood come from the liver, kidney, intestine, endocrine gland, and immune tissues. Okay. Then oxygen enters the blood in the lungs and leaves the blood as it flows through the tissue. Then carbon dioxide enters the blood from the tissues and leaves the blood as it flows through the lungs. I think uh, we are already aware of these points. We have discussed in the last semester as well. So whenever we inhale, we take more amount of oxygen. That oxygen is transferred to our blood. When the blood circulates our body, that oxygen is transferred to all the tissues which are present in our body. Then, with the help of capillaries, there is a gaseous exchange. So the gas which is present in the blood. So we have, where can I draw? Where can I draw? So here, so we have blood. It has oxygen. So here we have tissues. We have number of tissues. So when the blood reaches the tissue, the oxygen in the blood is transferred to the tissue. This tissue has some carbon dioxide or some waste. So those waste and the carbon dioxide gets transferred to the blood. Then again, this blood goes to the lungs where the carbon dioxide is sent out. And when the blood reaches the liver and kidney, the waste is sent out. So this is the cycle. Clear? Next is formed elements. So here, so the next one is the content which is present here, the 45% of formed elements. That's what we are going to discuss. Formed elements. So the RBC, red blood cell, WBC, white blood cell, and platelets. These are the formed elements. So the RBC is also called as erythrocyte or erythrocytes. It forms 95% of the formed elements. That is, the RBC is the majority of the content. Then the white blood cell, which is also called as leukocytes. Then platelets, they are also called as thrombocytes. So these two forms the remaining 5% of formed elements. So formed elements has only these three elements: RBC, WBC, and platelets, where RBC contributes to 95% and WBC and platelets contributes to the remaining 5%. Then we saw the other names. Or the other name for RBC is erythrocyte. The other name for WBC is leukocyte. The other name for platelet is thrombocytes. There is also another name for platelet that is called cell fragments. We generally don't use this term. We use either platelet or simply thrombocytes. Then this is additional information. Only the WBC has nucleus, while the RBC and platelet, they don't have any nucleus. Then the production of blood. The process of blood cell production is called as hematopoiesis. This is the process through which the blood is produced in our body. That process is called as hematopoiesis. So in fetus, what is the meaning of fetus? Anybody?
Unborn baby.
Yes, the unborn baby which is present inside the mother's body. It is still alive, but it is present inside the mother's body. So in the fetus, the hematopoiesis occur in liver, thymus, spleen, lymph nodes, and red bone marrow. So these are all the places where hematopoiesis occurs. That means these are all the places where the blood cells are produced in fetus. But once the baby comes out of the mother, that is after birth, hematopoiesis primarily occurs only in the red bone marrow. So this is the only place where the blood cells are produced after birth. So all the formed elements of blood are derived from a single population of stem cell called hemocytoblast. So I'm not sure how many of you still remember the term called precursor cell, progenitor cell, all those things. It is like a hierarchy, omnipotent, oligopotent, those things. So anyway, so we have a cell called as hemocytoblast. So this is the parent cell. We can call it as a parent cell, So from these cells, after so many differentiation, proliferation, we get the blood cells. So we will discuss that in the upcoming slides. So all the blood cells are derived from a single population of stem cell called as hemocytoblast. So this hemocytoblast, the parent cell, is a stem cell. So this hemocytoblast is located in our red bone marrow. Okay, so many texts, but anyway, let me try to explain it in simple terms. Hemocytoblasts are the precursor cells capable of dividing to produce daughter cells that can differentiate into various types of blood cells. Differentiate, it means one cell gets transformed into another cell. That is what we call it as differentiate. Proliferate means one cell gets divided into two cells. This is called as proliferation. Differentiation means we have one cell, it is transformed into another cell. So for example, initially it is WBC. After differentiation, it becomes RBC. So one cell gets totally transformed into another cell. That is called as differentiation. If one cell gets divided into two or multiple cells, that is called as proliferation. So anyway, hemocytes are precursor cells capable of dividing to produce daughter cells. So we have hemocytoblast, that is the parent cell or the precursor cell. It is capable of dividing to produce daughter cells. So once it is divided, it produces daughter cells. Those cells can differentiate into various types of blood cells. Either it could be the WBC, RBC, platelet, whatever it is. So next point, when the hemocytoblast divides, one daughter cell remain as a hemocytoblast. So we have the hemocytoblast, the precursor cell, it gets divided into two. Imagine this hemocytoblast is divided into two. So one cell still remains as hemocytoblast itself, while the other daughter cell differentiates to form one of two types of the intermediate stem cells. So I don't think I have a picture for this. Anyway, so imagine this is the precursor cell or parent cell, right? Then in between we have so many cells, and finally we have RBC or BC. So this is the precursor cell and this is the final cell. So the cells which are present in between these hierarchy, those are called as intermediate cells, intermediate stem cells. So this precursor cell, hemocytoblast, is a stem cell, and this cell either gets differentiated or proliferated and forms a series of cells. See, cell 1, cell 2, cell 3, cell 4, cell 5, and the cell 5 is the RBC, right? So when the hemocytoblast divides, one daughter cell remains the hemocytoblast itself. So here, one daughter cell, the first daughter cell remains hemocytoblast, while the other daughter cell differentiates to form one of two types of intermediate stem cells. So these are called as intermediate stem cells. So the cells which are intermediate, which are present in between the precursor cell and RBC cell. The steps which are present in between, those things are called as intermediate cells. So there are two types of intermediate stem cells. One is myeloid stem cell or lymphoid stem cell. So when this hemocytoblast divides into two, one remains as the hemocytoblast itself. The other one, it could be either myeloid stem cell or it could be lymphoid stem cell. Clear? So we have the hemocytoblast, which is the precursor cell. It is capable of dividing to produce daughter cells that can differentiate into various types of blood cells. Again, I'm repeating, differentiation, one cell transforming into another cell, that is called as differentiate. Proliferation means one cell gets divided into two or multiple number of cells. So here, differentiation. When the hemocytoblast divides, one daughter cell remain as the hemocytoblast itself. The other daughter cell will be either the myeloid stem cell or the lymphoid stem cell. So these two stem cells are again called as intermediate stem cells. So the red blood cells, platelets, and most of the white blood cells develop from the myeloid stem cell. So myeloid stem cell, it is responsible for the red blood cell, it is responsible for the platelet, and most of the white blood cells. Whereas the lymphoid stem cell, it is responsible for the production of lymphocytes. Is it clear? How the WBC platelets and white blood cells are developed? It starts from the hemocytoblast. This is the precursor cell. So this precursor cell gets differentiated into various types of cells. So this precursor cell goes for division, that is proliferation here. divides. So this precursor cell initially goes for proliferation, where it becomes two cells. One cell becomes two cells. This is the parent cell. These two are the daughter cells. Among the two daughter cells, one daughter cell remains the hemocytoblast itself, whereas the other daughter cell becomes the myeloid stem cell or lymphoid stem cell. If it is a myeloid stem cell, it can produce the WBC, RBC, and platelet. If it is the lymphoid stem cell, it can produce lymphocytes. Clear?
Clear.
Clear.
Okay. Then myeloid stem cells give rise to several intermediate cell types. Okay, these intermediates include the pro-erythroblasts which produce RBC. Okay, these are all just extra information. Myeloblasts which produce basophils, eosinophils, and neutrophils. Monoblasts which produce monocytes. So these are all again subgroups which are present in our blood. Apart from the WBC, RBC, platelets, these are all the other cells which are also present in our blood. That is, where is it? Where is it? Where is it? Basophil, eosinophil, neutrophil, monocytes, platelets we already saw. So these are also other cells which are present in our blood. So where is it? Okay, these intermediate produce pro-erythroblasts which produce RBC. Okay, fine. And yeah, here is the picture. I thought I didn't add it. Okay, anyway, so here we have the parent cell, the precursor cell, that is hemocytoblast, right? So this gets proliferated into either myeloid stem cell or lymphoid stem cell. If it is the lymphoid stem cell, it leads to production of lymphocyte. If it is a myeloid stem cell, it leads to the production of either RBC, platelet, or basophil, eosinophil, neutrophil, monocyte. Okay, so the cells which are present here, these things, these are called as intermediate. Where is it? Where is it? Here, this one, this intermediate stem cells. So the cells which are present between the hemocytoblast and the RBC or platelets, basophil, these cells, these things comes under the intermediate stem cell. Intermediate stem cell, right? It is like a process. Fine. Then next topic would be the hemostasis. Anybody know what is the meaning of hemostasis? Anyone?
The hardening of the blood,
Sorry?
the solidification or the oxidization of the blood, so that the blood clots up.
Ah, okay. Fine. Correct. We simply call it as blood clot, but the scientific name is hemostasis. So hemostasis is nothing but cessation of bleeding. So say for example, you are riding a motorcycle and you had a fall, you got injury and you got blood coming out of your body. If the blood keeps on coming out of your body, after some point of time, your brain will get a shock. Eventually, you will go to coma and you will die. So what happens is, once the blood is exposed to the atmosphere, hemostasis occurs. That is, blood clot happens. So because of the blood clot, we are alive, because it prevents the excessive blood flow away from, out of our body. So that is what we call as hemostasis. So that is nothing but cessation of bleeding. It is primarily required to maintain the homeostasis, the stability inside our body. So if there is no cessation, if the blood keeps on going out of our body, it will lead to low blood volume, because more amount of blood is leaking or more amount of blood is going out of our body. This will lead to low BP. Ultimately, it will lead to death. So in order to avoid death, we should have hemostasis. So hemostasis occurs in 3 stages. The first one is vascular spasm. Second is platelet plug formation. Third one is coagulation. So these are the 3 steps which occurs during hemostasis: vascular spasm, platelet plug formation, coagulation. So we are going to discuss about these 3 steps. First is vascular spasm. So I have mentioned it like how to say, like a sentence itself. Damage to blood vessels can activate the nervous system reflexes that cause vascular spasm. What do you mean by vascular spasm? Any idea?
Your blood vessels can contract and relax. Spasm.
Yes, exactly. So imagine this is our blood vessel. Normal time, this is how the blood vessel is. After going under vascular spasm, the diameter of the blood vessel will be, it goes small. This is called as vasoconstriction. C-O-N. Yes, it is very difficult to type with the mouse. Constriction. That diameter gets increased than the normal level, then it is called as dilatation. Vasodilatation. L-A dilatation. L-A-T-A-T-I-O-N. Anyway, so this is normal, the normal diameter of our blood vessel. If the blood vessel, the diameter of the blood vessel is increased, that is called as dilatation. If the blood vessel, the diameter of the blood vessel is decreased, that is called as constriction, vasoconstriction. So vascular spasm is nothing but vasoconstriction. So damage to the blood vessel activates our nervous system reflexes that causes vascular spasm, that is vasoconstriction. So vascular spasm is the immediate and temporary constriction of blood vessel. Constriction, this is what I mentioned, vasoconstriction. Vaso means blood vessels. It could be any blood vessel, either artery or vein or capillary or arteriole or venule, whatever it is. All those vessels comes under the term vaso. So vasoconstriction means reduction in the diameter of blood vessel. So vascular spasm is the immediate and temporary constriction. This is not a permanent constriction. It is just a temporary. So temporary constriction of a blood vessel. So constriction can close small vessels completely. If the blood vessel is already this much small, after vasoconstriction, it will be very, very small, which can totally cut the blood supply itself. So constriction can close small vessels completely and stop the flow of blood through them. So if the blood vessel is somewhat big, after vasoconstriction, still there will be a small amount of blood flow. But if the blood vessel is this much small in the beginning itself, then after vasoconstriction, it becomes very, very small, tiny. And hence there is no blood flow. So this is the first stage in hemostasis, vascular spasm. That is, the blood vessels goes for vasoconstriction. Next one is platelet plug formation. Again, I have so many texts, but once you read, it will be easier. It's simple. The platelet plug is accumulation of platelets that can seal small breaks in blood vessel. That is, this plug formation is nothing but we have multiple number of platelet cells which comes and settles down at the place of injury. That's it. To simply put, this is the simple explanation. The platelet cells which is present in our body comes and accumulated or settles down at the place of injury. So platelet plug is an accumulation of platelets that can seal small breaks in the blood vessels. The platelet plug is not same as a blood clot, but the formation of the platelet plug is an important step in the blood clot formation. So anyway, the plug formation is not exactly equal to the blood clot. It is somewhat different. The platelet plug formation is very important in maintaining the integrity of the circulatory system. It is self-explanatory. Small tears always occur in our blood vessels, that is smaller blood vessels and capillaries, many times each a day. And this platelet plug formation quickly closes them. So say for example, you are simply walking on an even surface. You saw a heavy rock on the way. So you try to lift it and throw it away. So what happens is you are bending down, you are lifting the heavy rock and your biceps muscle has to contract. So what happens is whenever you do that, the very, very tiny blood vessels present in the biceps, like capillaries, sometimes they may get damaged. So what happens is this platelet goes there and forms the platelet plug. So small tears occur in the smaller vessels and capillaries many times each day. So whenever we do regular activities, whenever we do some push-ups or sit-ups, whatever it is, there may be a small tear in the blood vessels. So this platelet plug formation quickly closes them or seals them. So people who lack the normal number of platelets tend to develop numerous small hemorrhages in their skin and internal organs. So for the people who have low number of platelets, because of these small tears, they may end up with hemorrhages. Hemorrhages means uh, bleeding, blood bleeding inside our body. That we call it as hemorrhages, right? So for people who have low number of platelets, they tend to develop multiple hemorrhages on their skin and internal organs. The last step is coagulation. So when a blood vessel is severely damaged, coagulation or blood clotting results in the formation of clot. The blood clot is a network of thread-like protein fibers called fibrin. So this is an important term, fibrin, that traps the blood cells, platelets in the fluid. So fibrin is nothing but a thread-like protein fibers. So imagine these are all fibrin threads. It will be like this, somewhat like this. So this is called as blood clot. So these fibers are activated and it will prevent the RBC cells passing beyond them. So it will just stop the RBC cells, WBC, platelet, all those things, right? So blood clot formation depends on next point. The blood clot formation depends on several clotting factors or coagulation factors. Normally, the clotting factors are in an inactive state and do not cause clotting. So the blood clot depends on multiple coagulation factors. These coagulation factors are normally in inactive state. That is, they are not active, but still it is present in our body. After injury, these clotting factors are activated. So once we get the injury, these coagulation factors are activated and hence this fibrin forms the trap, what to say, the clot. This is the pictorial representation. So these are all the fibers. So these yellow color string-like things are the fibers. We have the WBC, the trapped red blood cells. Platelets, it is not very clear here. But anyway, you will get you will get an impression. So this is how the blood clot look like. So we have the RBC cell. These yellow structures are the fibrin network. It's like catching a fish with a fish net, simply put. So the fish is the RBC cell, the fish net is the fibrin network, right? So this is about hemostasis. We saw vascular spasm, that is the blood vessel go for vasoconstriction. Then platelet plug formation, that is at the site of injury, platelets come there and settle down and form the plug. Finally, the coagulation factors are activated and hence the RBC, WBC platelets are trapped in the fibers. This is about hemostasis. Any doubts or questions here? In these steps, vascular spasm, platelet plug formation, coagulation. Any clarifications or questions? Is it clear? Can I proceed?
Clear.
Okay. Next is control of clot formation. We saw, where is it? Okay, we saw about the vascular spasm. We saw about the platelet plug formation. We saw about the coagulation. That is, we have the blood vessel where because of the coagulation factors, we have the fibers developed in the blood vessel and hence the WBC, RBC, platelets and other blood cells cannot pass through this clot. But after some times, eventually these cells will be able to pass. We are going to see how it happens. Control of clot formation. Again, we have multiple text, but it is very easier to understand. So, where is it? Okay, without control, clot formation would spread from the point of initiation through the entire circulatory system. So again, let me draw that. So imagine we have the blood vessel. Here we have the clot because the coagulation factors are activated. And here are the blood cells which are trapped, right? So here is the place of injury. And we have the clot formed over there. So if the coagulation factors are not controlled, these clot will be keep on spreading or developing throughout the blood vessels like this. And because of this, the person may die. And this should not be happening. So what happens is, to prevent the unwanted clotting, the blood contains several anticoagulants. So we saw coagulating factors which enhance, which initiates blood clot, right? Likewise, we also have another set of enzymes called as anticoagulants, which means this will stop the spread of clot. So to prevent the unwanted clotting, blood contains several anticoagulants. These anticoagulants prevent clotting factors from initiating the clot formation under normal concentration in the blood. At the site of injury, so many clotting factors are activated and the anticoagulants are unable to prevent the clot formation. However, away from the injury site, the activated clotting factors are diluted in the blood, anticoagulants neutralize them, and clotting is prevented. Can anyone explain these three sentences? Just read it once, you will get an idea. Try to explain these three sentences. Would anyone like to explain? Just read these three sentences.
Let me read it, Doctor. Can
Yes, yes, yes.
This anticoagulant prevent clotting factors from initiating clot formation under normal concentration in the blood. At the site of injury, so many clotting factors are activated that the anticoagulants are unable to prevent the clot formation. However, away from the injury site, the activated clotting factors are diluted in the blood. Anticoagulant neutralize them and clotting is prevented.
So what does it mean?
From the function of the
Coagulant.
coagulant prevent the unnecessary clotting can so that from initiating clot under a normal concentration in the blood. The second point is at the site of injuries that for example like a wound blood, doctor can wound, so the many clotting factor activated that the anticoagulated unable to prevent the clot formation. However, away from the injury site, the activated clotting factor are diluted in the blood, and the anticoagulant can neutralize them, and the clotting is prevented. So, if the injury, what we call activated clotting factor in the blood can
Uh-huh.
be um, uh, the activated clotting factor. Also, the activated clotting factor are in the diluted in the blood, so the anticoagulant can neutralize them, so the clotting is prevented.
Okay. Okay. Anyway, it's a good try. Good.
Is that correct or partially correct, Doctor?
Yes, yes, correct, but not as in a proper flow.
Okay.
Let me try to explain it. Wait.
Oh yes, the last point, Doctor.
How to erase? Okay, erase. Okay. So okay, so imagine this is the blood vessel, and inside the blood vessel we have blood which is flowing in this direction, right? The blood contains WBC, RBC, platelet, eosinophil, thrombocyte, erythrocyte, all those things. Along with those cells, we also have anticoagulants and coagulants. So along with the blood cells, we also have the coagulation enzyme and anticoagulation enzyme. So those things are also present in the blood. So imagine there is a small cut over here, right? So once there is a cut, these three steps immediately happen over there. So vascular spasm, platelet plug formation, coagulation. So this blood vessel somewhat becomes like this. I'm not able to draw it exactly, but just imagine. So here we have vasoconstriction. Imagine here the diameter is more, here the diameter is less, right? So there is a vasoconstriction. Then there is a plug formation, right? So here the blood cells, the WBC, RBC, those things are trapped. So once there is a cut, along with these three steps, the the coagulation enzyme that gets activated, right? So once those coagulation enzymes are activated, these three steps happen spontaneously, right? So okay, so there is a vasoconstriction, there is a plug formation, and we also have that fibers enzyme. Sorry, what is it? What fiber is it? Protein fiber. Yeah. So we have protein fiber inside to prevent blood leaking from here. Okay, next what happens is there is a clot formation, so the blood leakage is stopped. Next, this anticoagulants try to play a major role. What happens is at the site of injury, there will be more coagulants. So this anticoagulants will not be able to neutralize them. But later, that is over here, after or away from the place of injury, here we have more anticoagulants which will be able to control the coagulants. So imagine you are walking alone on a street and you come across a group of people. Suddenly the group of people are attacking you. You will not be able to tolerate them. You will not be able to respond them. So what happened? You run away from them, right? That's what we all do because we are alone. On front of us, there are a group of people who are suddenly attacking us. We will not be able to neutralize them. We fall back. But later, what you do? You call. You inform your friend that some people are attacking me. Try to come and help me. So again, you call your friends, more number of friends. You invite like 10 people. So along with the 10 people, you will be able to control those who initially attacked you, right? So imagine you are the anticoagulant. And so imagine you are here, you are the anticoagulant. And the group of people who are attacking you, they are like four members. They are the coagulants, right? So these four people are attacking you and you are not able to withstand. So you fall back. Later you call for support and suddenly you have 10 more people to defend you. So now you fight with them, and those people fall back. The same scenario is happening over here inside the blood vessel. So the blood, it contains both the coagulants and anticoagulants. So here, the pathway where you are walking, that is the blood vessel. So and the U or the anticoagulant, those four people, they are the coagulant. So when there is an injury, suddenly these four people, they try to work hard. So you as a single person cannot fight with them. Later, you call for a help, you get a help, and you try to neutralize them. So this is the factor happening inside a blood vessel as well. So these coagulants, so again, repeating from the beginning, without the control, the clot formation would spread from one point to the initiation to the entire circulatory system. Yes, if the clot is not controlled or not contained, that's the clot will spread throughout the blood vessel, throughout the circulatory system. So in order to prevent that, we need anticoagulant. So this anticoagulant is already present in our body, but at the time of injury, the coagulation factors, the coagulation enzyme, they try to work hard, they try to dominate. So these anticoagulants will not be able to withstand them. So hence there is a clot over here. But away from the site of injury, there will be more anticoagulants where they will be able to control the coagulants. Clear? So these anticoagulants prevent clotting factors from initiating the clot formation under normal concentration of the blood. At the site of injury, so many clotting factors are activated that the anticoagulants are unable to prevent the clot formation. So anticoagulants are here, here, but you are not able to control these four. However, away from the site of injury, that is, you are calling for help, the activated clotting factors are diluted in the blood, anticoagulants neutralize them, and the clotting is prevented. So you call for help, you get your friends come and they fight for you, and they neutralize these four people. That's it. So this is how the clot formation is under control. Clear? Any doubts? Or do you want me to repeat?
I think it's okay.
Okay. Okay, next, I think this is the last topic for today's lecture. It is about 23:50. Okay, so blood grouping. So I hope you all are aware of different types of blood groups. Can anyone mention those groups? Do you know our blood can be categorized into different groups? Can anyone mention
Blood
any?
A, B, AB, O positive, O negative, A positive, A negative, all those.
Yes, exactly. So those things comes under, where is it? Yeah, ABO blood group system. So this is a system, this is the thing which classifies the blood into A, B, AB, and O, right? So anyway, the surfaces of the red blood cells have a marker molecules called antigens. So every red blood cell has a molecule, has a marker kind of thing called antigen. Then the plasma contain proteins called antibodies, which bind to this antigens. So every red blood cell has a marker called antigen. Then the plasma which is present in the blood, they contain a protein called antibodies. So this antibody always tries to gets attached with the antigen, like North Pole and South Pole. We know that in physics they used to say opposite poles attract each other. Likewise, this antibody will always try to reach the antigens. Antibodies are very specific, meaning that each antibody can bind only to certain antigen. So there are different types of antigens like this, like this, like this, like this. and we have different types of antibodies like this, uh, like this. How to draw for this? Something like this and something like this. So these are all antigens. These are all antibodies. So antibodies are very specific, meaning that each antibody can bind only to certain antigen. So this antigen, yeah, this antigen will get attached only to this antibody. Sorry, yeah, antibody. It will not get attached to this or this. So they have very specific requirement. The ABO blood group system is used to categorize human blood based on the presence or absence of A and B antigen. So based on the presence of the antigen A and B, we divide the blood into multiple categories. So when the antibodies in the plasma bind to the antigen on the surface of the red blood cell, they form the bridge that connect the red blood cells called agglutination or clumping. That is how to mention, how to draw. When the antibodies in the blood bind with the antigens on the surface of the red blood cell, they form bridges. Mm, I have a picture. I will explain it over there. So it forms a bridge like this. So these are antigens. These are antibodies. Okay, so when they are attracted to each other, they form a clumping or agglutination. Do you know clump? Have you ever seen a video where there is a video in the, there are not one, there are multiple videos in YouTube where they explain what happens to the human blood when a snake bites us. Have you ever seen such kind of video?
Yes, doctor.
Yes. They will take the human blood from their vein, they pour it in a beaker, then they add the snake venom to the beaker and they mix it. What happens is this agglutination or clumping. The blood becomes like a jelly or like a jam, like a not syrup, but like a ketchup. Imagine something like that happening inside our body. So if the antigen and antibodies they bind, that's what happens. So anyway, these are the different types of blood. So we have type A, type B, type AB, type O. So type A blood has type A antigen, type B blood has type B antigen, type AB has both the A and B antigen, type O blood has no A or B antigen on the surface. So again repeating, blood group A has A antigen, blood group B has B antigen, blood group AB has both A and B antigen, blood group O does not have any antigen, that is antigen A or antigen B. So in addition to the type A and type B antigen in the group, there are also two types of antibodies associated with this blood group. That is anti-A antibody and anti-B antibody. So apart from these two antibodies, the plasma proteins has protein called, so sorry, the plasma protein has antibodies, right? So apart from those two, we also have another two antibodies, that is anti-A antibody and anti-B antibody. So anti-A antibodies act against A antigen, anti-B antibodies act against type B antigens. Plasma from type A blood contains anti-B bodies and plasma from type B blood contains anti-A bodies. So these points may be somewhat difficult to understand, but you try to keep reading it so you will be able to understand. Then type AB blood has neither type of antibody and type O blood has both anti-A and anti-B bodies. Do we have, okay, here is the picture. So these are the four different blood groups: group A, group B, group AB, group O. So these are the red blood cells and these are the plasma, that is the antibodies. Okay, let me explain here. So antigen A, this is how the antigen A looks like a small bubble. antigen B, they look like a small triangle. For blood group A and B, the RBC has both antigen A and antigen B, that is both the small balls and the small triangles. For blood group O, there are no antigen, so it remains flat. Then in the plasma, we have anti-B body. So can you see here, it is somewhat like a triangle. So these antibody will try to fit in over here. And here, this antibody, it has this kind of shape, which is capable of attaching with these balls, small molecules, right? Whereas for antigen A and B, sorry, whereas for the blood group type AB, there are no anti-A body or anti-B body. So here it is empty. It is difficult. Then for type O, sorry, for the blood group O, we have both the antibodies, anti-A antibody and anti-B antibody. So we have that this for the molecules, ball-like molecule, and this for the triangle-shaped antigens. So they are not supposed to get attached. What happens is, so imagine, so here we have type A RBC, then type B antibody. So here we have the ball-like structure, here we have triangle-like structure, and hence here there is no bonding, there is no agglutination. Whereas here we have again blood with the small molecules. Here we have anti-A antibody. Here it is anti-B antibody. Here it is anti-A antibody. But the donor remains the same. Do you know what is the meaning of donor and recipient? Donor is someone who gives, who donates. Recipient means someone who receives, right? So here, in both the scenario, we have type A blood, and here we have anti-A antibody. So what happens is this anti-A antibody tries to form a bond with this RBC. So this is agglutination. So this is not good. This is acceptable, no agglutination. So try to read this later. I will upload the slide today. So try to read this. If possible, try to read from the book as well, so that you will be able to understand. Or else the next class, the offline class, I will try to explain by drawing. So in online, I have difficulty in drawing. So maybe in the offline class, I can explain this once again, right? But anyway, as of now, just remember the types of blood and the, where is it? And the antigens and the antibodies that are present in the blood. That is enough. Maybe in the next class, I will try to explain it more. Okay, anyway, so I think this is the last slide. Yes. So a transfusion is the transfer of blood or blood components from one individual. So this is about the blood transfusion. So the transfusion is the transfer of blood or blood components from one individual to the other. Okay, self-explanatory. And infusion, on the other hand, is the introduction of fluid other than blood, such as the saline water or a glucose solution into the blood. You know what happens is when there is a need for a blood, that is when a person is suffering from a blood loss and he or she needs a blood. If there is excess blood in the blood bank, they can provide that blood. In some rare scenarios, instead of blood, doctors can inject this. Doctors can inject, where is it? Okay, so doctors can inject either a saline water or a glucose solution into the body. This is also acceptable way, but mostly they will go for the blood. In very rare scenarios, they will use the saline water or glucose solution. In many cases, the return of blood volume to the normal level is all that is necessary to prevent a shock. So what happens is, again, an example, a person is riding a motorcycle, he had a fall, he got head injury, so there is a heavy blood loss. So they take the person to the hospital. If the blood loss is beyond the threshold level, your brain will go for a shock. That means the person will enter the coma stage and eventually he will die. So in order to prevent that shock, sometimes in some very rare scenarios, in very urgent scenarios, they will inject the saline water or a glucose solution. After injecting that, the blood, the blood will be secreted from the bone marrow. Eventually, the body produces enough red blood cell. If the person survives, the bone marrow will start to produce red blood cells to replace that were lost. When a blood transfusion is performed, the donor is the person who gives the, this is what I explained earlier. The donor is the person who gives the blood. Recipient is the person who receives it. Anyway, so apart from using the saline or a glucose solution, we have A, we have B, we have AB, we have O. So if a person with the blood group A, who can give blood to this person? A person with A blood group needs blood transfusion. So who can donate their blood? Can anyone name it?
Blood O and A.
Yes. So people with the blood group A and O can donate the blood. For B, again, people with
B
blood
and
group
O.
B and O. For AB,
Only AB.
only AB. What about O?
Oh, I mean AB and O.
Universal.
A, B, and O. So that is why generally we call O blood group as universal donor, because that they can donate their blood to any people. What about O?
Only itself.
O.
O.
Yes. But again, this is what generally people tend to understand, which people are aware of. But still, there are many subdivisions even inside this, because there is something called as A positive, A negative, B positive, B negative, AB positive, AB negative. Okay, O positive, O negative. So anyway, we will discuss those things in the next class so that I can explain the blood grouping well better in the offline session. When is the next class? Do we have online or offline?
I think will be offline.
Today, where is it? Where is it? Where is it? Where is the calendar?
I think it's next week.
Today is 18th, right? 25th. Yes, it is going to be offline class. Okay, so then again, I will try to explain these things along with that positive, negative, what is the meaning of that and how that plays a role in blood transfusion. So anyway, as of now, today we discussed about the functions of the blood. We discussed about the components that are present in the blood. We discussed about the blood plasma, again, which is also comes under the composition of blood. Then the elements, other elements that are present in the blood. Then blood transfusion. So we saw the function, we saw the composition, we saw the centrifuge test. We saw the sub groups of the plasma protein, that is albumin, globulin, fibrinogen. Then the function of albumin to prevent the movement of water. Then globulin to protect against the microorganism. Fibrinogen to prevent, to form the blood clot. Then the solutes that are present in the plasma. Then the mechanism of how the blood work. Then the elements, formed the elements, how the blood is formed, I mean how the blood cells are formed. Then hemostasis, how the hemostasis occurs by the vascular spasm, platelet plug formation, coagulation. Then how the clot formation is kept under control. Then blood grouping. So next class, I will try to explain again the blood grouping along with the factors positive and negative, A positive, A negative, B positive, B negative, all those things, right? So as of now, do you have any questions? Do you have any clarifications? whether you understood or you didn't understand anything. Anyone?
Understood, doctor.
So far, so good. Okay. So from next class onwards, that is from next class onwards, we are having offline class, right? So every class we will have stop presenting. Okay, so from next class onwards, we will have an activity in every class. By the end of the session, we will have an activity so that I want the students to participate more, not like the last semester. Only few students kept on participating, only few students performed well, whereas rest of the students were like on the average level. So in order to avoid that and to improve your participation, from next class onwards, there will be an activity. Since we have a 3 hours of class, maybe I can take a lecture for 2 hours or 2.5 hours. The last half an hour is for your activity. So we will perform some activity, any activity related to the topic. And those who perform well will be maintaining a note, a log note, so that by the end of this semester, if there is a need, I can provide some extra marks to them because they have actively participated throughout the semester. So that's the concept behind this. Clear? So next class onwards, there will be an activity. I want everyone to participate, not just one or two. I want everyone to participate so that it will be helpful for me and as well as you at the end of the semester. Clear?
Clear.
All right, doctor.
Right. So we can wind off with this. I will try to mark your attendance manually so that you can check later whether your attendance is being marked or not. If your attendance is not marked, do let me know in the next class so that I can mark it again. Okay. Thank you. See you all next week. And happy Hari Raya.
Thank you,
Thank you,
doctor.
doctor.
Thank you, doctor.
Thank you, doctor.
Thank you, doctor.
Selamat Raya.
Thank you, doctor.
Thank you. Thank you. Thank you. Thank you.
Thank you, doctor.
Thank you.